The overall goal of the following experiments is to study disease-associated mitochondrial phenotypes in human fibroblasts using a live cell imaging technique. This is achieved by first obtaining a skin biopsy and preparing it for tissue culture. First, human fibroblasts will propagate from the skin sample within the first 2 weeks. Next, the fibroblasts are detached and seeded into chambered cover glass wells. After 24 to 48 hours, live cell imaging dyes are applied to evaluate mitochondrial function, morphology, or mitophagy by live cell imaging technique. Ultimately, software-based analysis of the images can reveal morphological and functional mitochondrial parameters in living human fibroblasts.
The major advantage of using primary fibroblasts from patients carrying disease-associated mutations compared to already existing cell models like tumor cell lines is that fibroblasts reflect important pathological features of the human disease without previous genetic manipulation based on endogenous protein levels.
These techniques can help to identify early signs of Parkinson's disease, such as alterations in mitochondrial function and dynamics, and to define new therapeutic strategies.
Begin with a fresh tissue biopsy, which has been taken by a biopsy punch. Place the specimen into a first dish.